Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on cerebrospinal fluid. Learning objectives, what we are going to discuss in this lecture. First of all, we are going to discuss the functions of cerebrospinal fluid. What role the cerebrospinal fluid is playing for our brain and spinal cord. Then we are going to trace the circulation path of cerebrospinal fluid. Then we are going to discuss about how this cerebrospinal fluid is formed, from where it is originated, and finally we are going to discuss about the removal of cerebrospinal fluid. Now, before we start this lecture, I would like to show you this figure: circulation of cerebrospinal fluid. Here you can see the brain is shown, and in the see they are showing the subarachnoid space, and with the white arrow, this white arrow this is the circulation of cerebrospinal fluid. So you see this figure and you will have idea that how see around our brain and spinal cord subarachnoid space is there, and in that the the cerebrospinal fluid is circulating. Okay, so this is the figure this way. Okay, so cerebrospinal fluid, in short, CSF. Now, what is CSF? CSF is a clear, colorless liquid, and this CSF circulates within the ventricles and in subarachnoid space, which is present around the brain and spinal cord. Now, what function this CSF, what is the function of this CSF? So CSF bars the exposed surface of central nervous system. Central nervous system meaning the brain and spinal cord. So the brain and spinal cord, the neurons of brains and spinal cord, they are in direct contact with cerebrospinal fluid, and this CSF completely surrounds our brain and spinal cord. CSF performs some important function, so we are going to discuss that functions also. Now, cerebrospinal fluid, where it is present, so CSF is found in and around brain and spinal cord, and this CSF forms liquid cushion, and as this CSF is acting as a cushion, it gives buoyancy to CNS structure. It provides buoyancy to our brain and spinal cord. Now, what is the meaning providing buoyancy to CNS structure? See, the buoyancy function of cerebrospinal fluid. Now, our brain floats within CSF and thereby reducing its apparent weight by more than 95%. So as the CSF is present around our brain, what happened? The actual weight of our brain is reducing. So this helps, this prevents this reduction of weight of our brain, help us help us to the brain being crushed under its own weight. So if we if we let's say if CSF is absent, then what happened? So that time our brain will be very heavier, and there are chances that due to its own weight the brain being crushed. Okay. Now one more thing, if CSF without the CSF support, it is also possible that the portion of the brain would sink into the foramen magnum. Some part of the brain will enter into foramen magnum. Okay. Foramen magnum is the opening, okay, for spinal cord, and it joins with medulla oblongata. So it is possible that the part of brain can enter into this foramen magnum if CSF is absent. Okay. So this is the buoyancy function of CSF. Now the fourth point, protection function of CSF. See, CSF provides a liquid cushion to delicate structures, that is our brain and spinal cord. They are protected by CSF because CSF is acting like a liquid cushion. And when we are doing sudden movement, let's say we stand up and we are walking, and or we are running, so that time it is possible that uh, our brain get it is possible that our brain can get injury by striking the inner wall of our skull. But as the CSF is present around the brain, so this jerking or the shocks are absorbed in CSF. So this way CSF is protecting our brain and spinal cord. Okay. So see, when you try to walk quickly in swimming pool, your movement are slowed as the water act as a movement buffer. So imagine you are in the swimming pool and the water of swimming pool. Okay. So due to the water of swimming pool, you cannot move, you cannot walk very fast in the water of swimming pool. Similarly, as we are walking or as we are running, what happen, if as I told you earlier, if CSF is not there, it is possible that the brain or the spinal cord get injuries, okay, by striking with the wall of skull or the vertebral column. But what happens, CSF is there, so CSF, this is the it is acting as the protective liquid. Okay, it is acting as a protecting cushion, and it is absorbing the shocks or the jerks. Okay. So in that way it is protecting the brain and spinal cord. Then see, CSF likewise help slow movement of the brain if the skull or body moves suddenly and forcefully. So suddenly, let's say a person experience an accident. So that time, this CSF is going to absorb the blow of the accident situation. Okay, so that way it will protect the brain and spinal cord. So CSF is protecting our brain and spinal cord. Then fifth point, environmental stability. See, CSF transport nutrients and chemical messenger to the brain and removes. So it is CSF nourishes our brain cell, and at the same time CSF removes waste product from the brain. Okay, so the neurons of our brain, they discard the waste product, they throw the waste product into the CSF. Okay. Now additionally, CSF protect nervous tissue from chemical fluctuations that would disrupt the neuron function. So CSF is protecting our nerve cell. If any chemical fluctuations are going to occur, so this CSF is protecting our neurons. Then the waste product which is produced by the neurons, okay, so waste product and excessive CSF, they are finally eventually they are transported into the venous circulation, that means the venous. So CSF, after completing its job, then it is going to absorb, it is going to enter into the venous blood, and this way the CSF enters into our venous circulation. Now the sixth point. Additionally, our brain is having, even though our brain is having a rich blood supply, CSF is nourishing the brain, and there are some evidence that the CSF carries chemical signal, the chemical signal in the form of hormones, and there are some molecules present in the CSF which induces sleep, and some molecules are responsible for our appetite, that means for our hungerness. Okay. So CSF is carrying this chemical signal from one part to the another part of the brain. Now CSF is a watery broth for our brain and spinal cord. Now, what is first? Let's understand broth. What is broth? See, broth is a savory liquid, and how this savory liquid is made. So see, in a container, water is boiled, okay, and in this boiled water, some bones, meat, and fish are added, and all this food boiled, okay, together, and then some vegetables are added to it. So the soup that is made, okay, of all these items, bones, meat, fish, and vegetable, that liquid, that savory liquid or the soup is known as broth, and it can be eaten alone, but this broth is commonly used to prepare other dishes such as soup, gravies, and sauces. But uh, here this is not the same broth we are discussing. This is the dictionary meaning of broth, but here we can understand that the CSF broth means it is nourishing the nerve cell of the brain and spinal cord. That's why they have used the term broth. Okay, so CSF is a watery broth, and the composition of CSF is similar to blood plasma, and from blood plasma this CSF is formed. Okay, so CSF is formed from the blood plasma, it comes out from the blood plasma. See, when we see the composition of CSF, so CSF contains less protein than the plasma, and the ionic concentration are different. So CSF's ionic concentration is different when we compare it with the blood plasma. For example, CSF contains more Na plus, Cl minus, and H plus than the blood plasma, and CSF is having less Ca twice plus and K plus in comparison of blood plasma. Okay, so in the seventh point we have discussed about the composition of CSF, what is CSF and how it is produced, and which are the ions that are present in CSF. Now the eighth point we are discussing. Now in this point we are discussing the formation of CSF. Okay, how CSF is originating. So see here, see this is the this red in color, is choroid plexus, and this choroid plexus here also. Okay, so this is in the lateral ventricle. Okay, then this is also choroid plexus, and this is also choroid plexus. So in each ventricle, in each ventricle, choroid plexus is present. So the choroid plexus hangs from the roof of each ventricle, and this choroid plexus forms CSF. Now what is choroid plexus? Okay, what choroid plexus is made up of? So this choroid plexus are frond shaped cluster of broad thin wall capillaries. So we can say choroid plexus is actually a cluster of blood capillary wall are thin, and this capillary are the broader one, and this cluster of capillary is known as choroid plexus. And this capillary network or the cluster, first these capillaries, they are enclosed by pia mater, and then cellular layer is present, and this cellular layer is of ependymal cell, and this ependymal cell, they line the ventricles. So in the figure we are going to see this entire setup. So here you see this one is this red in color is the choroid plexus, and we are now zooming one section. Here in this figure they have shown the one section. See over here it is written section of, this is not the entire choroid plexus but the part of a choroid plexus. So see in the structure of choroid plexus, in the center, as you can see, connective tissue of pia mater they are showing. Okay, this is the connective tissue of pia mater, and here also the pia mater is present, and this red in color, this one is the blood capillary. So capillary, then outside this, these are the ependymal cell. So you can imagine this all this, what is this part, this entire portion, which part is this? Okay, so this is the cavity, this we can say this is the cavity of ventricle. So here they are writing cavity of ventricle, and now from this capillary of choroid plexus, what is happening? See, from this capillary, CSF, cerebrospinal fluid. Okay, so the blood is present in capillary, and the blood from blood plasma, CSF is coming out. Okay, where it is coming out? So the CSF is entering into the cavity of ventricle. Now CSF CSF forms CSF forms as a filtrate, and in this CSF glucose is present, oxygen is present, vitamins are present, and ions are present. For example, Na plus is there, Cl minus is there, Mg twice plus is there in the CSF. Okay, and and from the CSF, okay, what is happening? Waste and unnecessary solutes or the ions, they are absorbed, they are returning back from CSF to the blood of capillary. Okay, so I'm showing you this figure. Okay, one more figure I would like to show you. For example, in this figure, okay, see, this is the lateral ventricle. Now let me zoom this. Okay, zoom. Okay, so see, lateral ventricle they are showing. This is the lateral ventricle, and in that, this choroid plexus is present. This is the choroid plexus, and this one, choroid plexus. And now we are zooming one section of choroid plexus. So see, first, this red in color, see, blood capillary, of choroid plexus. Okay, first of all, then in the capillary wall, see, endothelium cells they are showing. These are the endothelium cell. Okay, then pia matter. So this one is the pia matter they are showing. Then you see these are the ependymal cell. Okay, now see, over here what is happening? This CSF is coming out. Okay, and here this part is the ventricle. So sure. Okay, now the ninth point. See, so the capillary that we have seen of choroid plexus, these capillaries are fairly permeable, and from this capillary tissue fluid filters continuously from the blood stream. Okay, now between the cells, ependymal cell, tight junctions are present. So the ependymal cells of choroid plexus, they are joined each other by tight junctions. Even though there are the presence of tight junction, still from this plasma membrane of ependymal cell, the ion pump or the ion channels are present. First of all, let me show you this on this figure. See, over here in this way. See. Okay, so this is the tight junction, okay, between the this is also the tight junction they are showing. Okay, so the plasma membrane at tight junction, in that the ion channels are present, and this ion channel or the ion pumps are selectively permeable. Okay, and these pumps, they are actively transporting certain ion. Actively means the use of ATP. Energy from ATP is utilized, okay, for the transport of ions. So now these ions from blood, they enter into the cavity of ventricle, and we know uh, these ions are the part of CSF pool. Okay, so this is how the ions are entering in the CSF. Okay, then uh, if we observe the if we see the composition of CSF, so we will find that CSF also mixes with the extracellular fluid, and this CSF mixes with extracellular fluid, and together both of them, they are bathing the neurons of brain and spinal cord. Okay, then now as from the capillary blood, ions are entering into the CSF. What happened? Ionic gradient is developed, and due to this ionic gradient, water from the capillary blood diffuses into the ventricle. Okay, so in short, between the ependymal cell, tight junctions are present. In the plasma membrane, there are the ion channel or ion pumps are present. This ion channel or ion pump, they are pumping or they are transporting ion, and in this is the active transport process. ATP energy is utilized. Okay, this ion enter into the cavity of ventricle, or this, we can say these ions are entering into the cerebrospinal fluid. Okay, now as the ions are entering into the ventricle or the CSF, what happened? Water also diffuse from blood capillary, and water enters into the cavity of ventricle. Okay, now in adults, the total CSF volume of about 150 ml, that is about half a cup, is replaced every 8 hours or so. So every 8 hours, the CSF which is circulating is replaced. Okay, so the volume 150 ml is replaced every 8 hours, and about 500 ml of CSF is formed daily. Now, this choroid plexus, another function of this choroid plexus also help to clean the CSF. And how this choroid plexus is cleaning the CSF? So it cleans by removing waste product and unnecessary solute, unnecessary ion and waste products are removed from CSF, and this waste product and unnecessary ions, they enter into the capillary of choroid plexus. So here you can see from CSF, waste and unnecessary solute, unnecessary ions, they are entering into the capillary of choroid plexus. Now here I am showing you one concept map, and in this concept map they have shown us the formation, circulation and absorption of cerebrospinal fluid. So see, first of all, see, you see this from heart. Okay, now from heart there arise aorta, from aorta arteries. Okay, so in the artery there is arterial blood, red in color. Okay, now where this arterial blood is going, okay, so here artery, they are going to reach into the brain and spinal cord. So the branch of artery, arteriole, then the branch of arteriole. Okay, so network of capillary, and that network of capillary here in the lateral ventricle or rest of the ventricle is known as choroid plexus. So now in the capillary of choroid plexus, lateral ventricle, so what happened, from blood plasma CSF is coming out from the capillary. Okay, and where it is entering, so now CSF is entering into the lateral ventricle. Now this, from lateral ventricle, this CSF through the interventricular foramina, this CSF enters into the third ventricle. Now arterial blood reaches to third ventricle's choroid plexus. So third ventricle's choroid plexus also produces CSF. This CSF is also entering into third ventricle. Now from third ventricle, CSF CSF aqueduct through aqueduct CSF enters into the fourth ventricle. Now here fourth ventricle's choroid plexus, from that CSF enters into the fourth ventricle. Now CSF from fourth ventricle through lateral and median apertures. Okay, from fourth ventricle CSF through lateral and median aperture, for this CSF now enters into the subarachnoid space, the space which is present around the brain and spinal cord. Now from subarachnoid space, this CSF finally reaches to arachnoid villi of dural venous sinuses. Okay, and in this sinuses venous blood is there. So now CSF is absorbed by arachnoid villi, and now this way CSF is entering, is mixing with the venous blood, and this venous blood is finally reaching to heart and lungs. Okay, so I'm showing you this concept map. Okay, this one. One more figure I'm showing you. See, in this figure, step by step, we are going to learn how the CF cerebrospinal fluid is produced and how it is circulated in our brain and also in spinal cord. So point number one, see, point number one. Okay, see this one, this red in color, this part. Okay, what is this? This is the choroid plexus. Okay, this one is the choroid plexus, which is present in each lateral ventricle, and this choroid plexus is secreting CSF. So with this arrow, as you can see from the choroid plexus, CSF is entering into the lateral ventricle. Now where this CSF is going, so now CSF, as you can see now, CSF, this part, okay, this is this part. So what is this? This is interventricular. Inter means in between. Which ventricle we are talking about? Lateral ventricle and third ventricle. So two lateral ventricles are there, and one third ventricle. So between two lateral ventricle and third ventricle, this aperture is known as the interventricular foramina, this part. Okay, so now CSF from lateral ventricle enters into the third ventricle. Now from third ventricle, see, third ventricle, in the third ventricle, so see here also in the third ventricle choroid plexus is present. So this is also, this plexus is also producing CSF. Okay, so now here in the third ventricle more CSF is produced. Now where CSF is entering, so see point 4. Now aqueduct, where aqueduct is present, see, cerebral aqueduct. This one is the fourth ventricle, this one is the third ventricle. So we can say between third and fourth ventricle cerebral aqueduct is present. So now what is happening? CSF flows down cerebral aqueduct, and now CSF is entering into the fourth ventricle. CSF is entering into the fourth ventricle. Okay, then now what is happening? See, CSF flows out two lateral aperture and one median aperture. So see here, but before that, see this point. This point, let me discuss first this point. Choroid plexus in fourth ventricle. See here, fourth ventricle, this one is the fourth ventricle. So this fourth ventricles choroid plexus also producing CSF. So this is the CSF they are showing. Okay, now where this CSF is going, so this aperture and this aperture. Okay, so there are two lateral aperture and there is one median aperture. So from fourth ventricle, now the CSF enters into and cross this lateral aperture, and CSF also cross this median aperture. After crossing this aperture, lateral aperture and medial aperture, this CSF enters into subarachnoid space. So this is the subarachnoid space of spinal cord, and here you can see this is the subarachnoid space. This one is all, these are the all subarachnoid space of our brain. So now CSF is circulating in the subarachnoid space. Okay, and now what happens? See, with the arrow they are showing CSF, and finally CSF reach to CSF reaches to arachnoid villus. Okay, so now what happens? So see, at arachnoid villi, CSF is reabsorbed into venous blood of dural venous sinuses. So let me show you that. One minute. In the figure I will show you arachnoid will lie. This is okay. Now in this figure you see this is the in the subarachnoid CSF is flowing. Now you see this part, see arachnoid villi, okay, and now we are zooming this arachnoid villus. So this is, see this part, this blue color, okay, this, what is this? Superior sagittal sinus or dural venous sinus, okay. And now CSF CSF passes to this arachnoid villus, okay, and then CSF is entering into the venous blood of dural venous sinus, okay. This is how the CSF is removed, okay. So here with the arrow they are writing CSF flow, okay. This is the arachnoid villus, okay. uh, and now we have completed the discussion of all the theory points, but I would like to show you all the figures one by one. This figure we have seen, okay, then this we have seen, and this figure, okay. See, key path of CSF venous blood, okay. And then this one. And this figure, okay. The flow of cerebrospinal fluid. So with this we have completed this lecture on cerebrospinal fluid. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kushti sir. I am from Ahmedabad, India. Bye bye. Namaste.